We're going to take a look at the metabolism of proteins. So how do amino acids enter cells? So remember that amino acids are hydrophilic and they cannot diffuse across the cell membrane, so they're going to need the help of a transport protein to get inside the body cells. So there's a couple ways that they can get in, but for right now we're just going to put down that they enter into the body cells by using a transport protein. Right, protein catabolism, so breaking down proteins. So we've got two steps here. So the first step is called proteolysis. So if we take a look at the word, proteo is referring to proteins and lysis is to split or break apart. And in proteolysis, all we're going to do is we're going to take the protein and break the peptide bonds and separate the amino acids. So if we've got this protein over here, we're just going to chop the peptide bonds holding the individual amino acids together and just separate these amino acids so now they're all individual. So that's proteolysis. And then the next step is deamination. So remember, D means to remove. And what we're going to be removing is the amino group from the amino acid. So here's a picture of an amino acid. And here's the amino group with that nitrogen there. And that amino group we can't do anything with. So we're just going to chop it off and throw it away. Okay, that's deamination. So we just take that amino acid, chop off the amino group, and we're going to take the amino group, and the liver is going to convert that amino group into urea. And then the urea will go through the blood to the kidneys and get filtered out into the urine and then excreted. And then what's left of the amino acid is called a keto acid. I won't ask you about that. You don't have to memorize a keto acid, but just this little carbon skeleton that's left of the amino acid, that gets sent into the Krebs cycle, part of aerobic respiration, and then we can just make ATP from that. Okay, another way we can deal with the amino group on the amino acid is called transamination. So instead of just chopping off the amino group and throwing it away, transamination, we're going to chop the amino group off, but we're going to reuse it. And we're going to reuse it to make a different amino acid, one that's not essential. So it's just an amino acid that we can make in our cells that we don't have to get from our diet. That's a non-essential amino acid. All right, so let's just show you the difference between deamination and transamination. So don't get upset about these pictures. I just want you just to understand just the basic idea of what's going on. Okay, so just to understand these definitions. So with deamination, remember D means to remove. So all we're going to do is we've got this amino acid. We're just going to chop off that amino group and throw it away. Okay, so that amino group gets chopped off, sent to the liver. Liver converts the amino group to urea, and that gets filtered out by the kidneys and excreted in the urine. And then what's left of the amino acid, this little carbon skeleton, that's called a keto acid, which you don't need to know, but that just gets fed into aerobic respiration and gets turned into ATP. Okay, transamination, a little bit different. What we're going to do here is we're going to chop off the amino group from the amino acid, but instead of throwing it away, we're just going to reuse it and make a different amino acid. Okay, that's all we're going to do. So we're just going to take this amino group, chop it off one amino acid, and we're going to put it on to this keto acid over here, and we end up making a different amino acid. Okay, so that's all we do with transamination. Okay, so don't freak out about this picture, bear with me. So let's look at deamination first. So we've got this amino acid here, glutamic acid in green. And remember, deamination, D means to remove, so we're just going to chop off this amino group from this amino acid and throw away the amino group. So the amino group goes to the liver, gets turned into urea, and we excrete that. And then what's left, this keto acid, will send that into aerobic respiration and we'll make some ATP from that. All right, so that's deamination. All right, transamination. Okay, what we're doing in transamination is we're going to chop the amino group off and reuse it and make a different amino acid. So we're going to start with this amino acid here. We're going to chop the amino group off. We're going to put it onto this keto acid here. And then we end up making this different amino acid up here in green. Okay, so we've just reused this amino group. We chopped it off this amino acid, put it over here, and made a different amino acid. Right? So this is a way that our body can make some different non-essential amino acids. So we don't have to get all of the amino acids from our diet. There are some that are non-essential that our cells can actually make, and we don't have to get it from our diet. Okay, so let's take a look at protein metabolism. And we've got some carbohydrates. And fatty acids. Both of those can be broken down into keto acids, which remember, you don't have to memorize keto acids, but just remember that carbohydrates and fatty acids, we can actually use those to produce these non-essential amino acids. So these are just amino acids that our cells can make on their own. You don't have to actually get them from your diet. And how we make these non-essential amino acids are by those transamination reactions. Okay, but there are 8 essential amino acids that our cells cannot make. And we have to get these from our diet. Okay, and some examples of essential amino acids that you do not have to memorize, but these would be things like methionine, tryptophan, leucine, and there's 8 of them total. Okay, so if you have some extra amino acids, so maybe you ate too many proteins for lunch or something, guess what we can convert those into? Store them for later. We can certainly convert them into fats, so some triglycerides. And we can also convert them into glucose. And then we can also use amino acids to produce a number of other things that are pretty important in the body. So we can produce purines and pyrimidines, and those are part of nucleotides. So those are the nitrogenous bases, the A's, T's, C's, and G's of the DNA and RNA. We can also use certain amino acids to make neurotransmitters like dopamine and norepinephrine, and also certain hormones, epinephrine and thyroid hormones. And then we can also use amino acids to make histamine, which is involved in inflammation and allergies. So here's that scary picture again from chapter 3. So this is just showing you that we can actually take some carbohydrates. So maybe we've got some glucose here, and we can work our way over and we can make amino acids from those. And we can also work our way over from the fats and make amino acids from those as well. Okay, so we can make these non-essential amino acids from carbohydrates and these fatty acids. So we can work our way over there. But the essential amino acids, those are the ones that our body can't make and you've got to get from your diet. And also note that these reactions are reversible. So if you do eat too many proteins, you can certainly store the extra proteins and work your way over and store those as fat. Or you could take the extra amino acids and you can make some new glucose with those if you needed to. So remember, that's gluconeogenesis. Okay, and then you don't need to memorize this either. This is just showing you the amino acid tyrosine. And from this amino acid tyrosine, we can make the thyroid hormones in the thyroid gland. And then depending on the cell, which enzyme they have, we can make the neurotransmitters dopamine and norepinephrine. And then in the adrenal glands, we can also make the hormone epinephrine. Okay, and then just kind of a summary of these catabolic pathways, breaking down these 3 nutrients from your diet. So looking at the proteins we just talked about, proteolysis. So remember, that's where you take the protein, break it apart into the individual amino acids. And then deamination is where you take the amino group off the amino acid and you throw it away, convert it into the urea. And then what's left of the amino acid you can feed into aerobic respiration and make ATP. And then we've got the triglycerides, breaking down the triglyceride into the glycerol and fatty acid chains, that's lipolysis. And then the fatty acid chains, breaking it into the little 2 carbon pieces into acetyl-CoA, that's called beta oxidation. And then glucose, splitting it in half, that first step of aerobic respiration, remember, that's called glycolysis. And then just remember that a lot of these pathways are reversible. So the red pathways are catabolic, breaking things into smaller pieces. And then the blue pathways are anabolic, you're making these larger molecules. And so you can see how you can work your way over, and if you eat too many carbs, you can certainly work your way over and store those as triglycerides, and so there's all kinds of ways you can go here.